Hi, everybody. Welcome to part 3 of this chapter. This part is going to be mostly about cell, cell division, and it's going to be pretty short, I hope. I know I've been pushing almost 30 minutes in my other videos, so I'm hoping this one's only 30 slides, but it's a good chunk of information. So let's get started. Cell division is a form of basically cell cloning. It helps to increase cell number and maintain the number of cells needed for a tissue. It does have a lifespan, however, and apoptosis is the name of cell death, and it's often controlled by the cell itself. So the cell cycle is what regulates cell division. Two types of cell division, mitosis and meiosis. When we talk about the cell cycle, we're talking about mitosis. We're talking about producing our egg and sperm. We're talking about meiosis. So mitosis, basically a clone. You have an original cell, you double everything inside the nucleus and all the organelles, and then you split it. The division produces two daughter cells, which are identical. Interphase is the non-dividing period. And so there's normal activities until the cell gets a signal to actually start dividing. And the first thing that happens is we have to double everything. And one of the things that's doubled is DNA. So this is showing you DNA replication here. Cell division is then the mitotic phase, the M phase, and it ends with cytokinesis. This literally means the division or splitting of the cytoplasm. So some details, interphase. So we're talking about somatic cells dividing here. Somatic cells are any body cells, but not the cells, the tissues that produce sperm and egg. So somatic cells, are mostly spending their time in interphase. 90% of all cells, if you were to take a sample of somatic cells, they're in interphase. So we have this normal phase. This G phase where they just kind of sit around. Stem cells are kind of unique and very valuable because they never enter this phase, they just continually divide. And some of the cells, like embryonic stem cells, are valuable because they're not differentiated on top of that. So that's why, like, if you've ever heard of someone harvesting an umbilical cord, that's because there's embryonic stem cells in there. Okay, so 3 phases of interphase if they are starting to divide. G1, which stands for gap1, this is the normal cell functions, but duplication of the organelles starts to occur. S phase stands for synthesis, and this is when the DNA is going to duplicate. G2 or gap2 phase, this is kind of this last slice here, and then this is kind of the last minute cleanup. So protein synthesis, if we need to double proteins, we need to double the centrioles. And then you can see here where the M phases. DNA replication, this is during the S phase. We have 2 enzymes that are very important, and we know they're enzymes because of that ASE ending. DNA helicase is, if you kind of see that hela, meaning like helix, so this actually unwinds the helix. It unwinds the 2 DNA strands and it disrupts those bonds. And then DNA polymerase is what bonds to the exposed bases, and it actually brings in new bases, and so it's linking new complementary nucleotides. It also acts as a kind of a spell checker to make sure those nucleotides are correct. Polymerase will only work in one direction, so while both strands are being replicated, only one side can actually form the leading strand. Only one side can go in like one continuous direction. The other is called the lagging strand, and this has to do it. So if DNA replication is overall left to right, what the lagging strand is going to do is it's going to go back, make a chunk, okay, a chunk of the DNA, and then it's going to kind of move on and make another chunk, move on and make another chunk, okay, because that DNA polymerase can only go in one direction. So one of the strands is leading and the other one is lagging. And then of course, you've got two identical DNA strands at the end. And this is a nice picture of DNA replication and of all of the enzymes that are involved. I'm going to fix this. I don't think this works. So I'll find another DNA replication video to put on here. And I don't think I fixed the other one either. So when you are looking at these slides, there'll be a different video here. Mitosis or M phase, this includes mitosis, which is prophase, metaphase, anaphase, and telophase, and then cytokinesis, the division of the cytoplasm. We're going to hit a lot of this in lab. We have a day that we're going to do all the phases. So interphase, the DNA is replicated. It's that G1, S, and G2 stuff there. The chromatin is loosely coiled, so you can't really see the chromosomes because they haven't thickened and doubled yet. Prophase, the chromosomes have thickened. They've coiled and they're visible. The centrioles are replicated and they start to kind of migrate toward the poles. And remember, this is a three-dimensional object, just like the globe, so we're using north and south just to indicate opposite poles. The centrioles move to the poles. There are astral rays which extend from the centrioles and it forms this nice little kind of star-like. Spindle fibers are part of the cytoskeleton and they help to interconnect the pairs. The chromatid, each copy kind of lines up, and the centromere is the center. Let me go back. Each copy of the chromosome is called a chromatid. Two sister chromatids are attached at a centromere. Okay, so it looks like a little X. The kinetochore is the area by the centromere where the spindle fibers, which is that cytoskeleton, which has helped support this whole process, attaches. Metaphase is when they're on the equator. So you got the two poles, the equator is in the center, they're lined up, ready to divide. Anaphase is when they actually split, so the sister chromatids are split apart, and it looks very nice in these pictures. Telophase is when you're starting to see the cytoplasm divide, although cytokinesis is after the whole thing has actually formed two cells. It looks like two bubbles, but you can clearly see some of the elements of the plasma membrane lined up here. The kind of bubble that's formed, or the indentation of the bubble that's formed, is called the cleavage furrow. And then cytokinesis is, when you have that cleavage furrow, it divides. Some people, some textbooks, some teachers will say cytokinesis is its own step. Some people will say it's telophase and cytokinesis. Whatever, you know what's happening. So here's the overall process of mitosis. It's nice and pretty. You've got them lining up here at the metaphase, splitting apart, and then you have two completely identical daughter cells. I did add the phases of mitosis here to this slide, so I guess I did change this one out. And this is a Bozeman video. It's nice. I think Khan Academy has a mitosis video as well as the Amoeba Sisters. So all of those are really good ideas. And if you've joined my playlist or saved my playlist, a bunch of them are on there. Okay, so now let's talk about some mistakes in the cell cycle. Cancer is a mistake in the cell cycle, and it's a disruption of cell division. The disruption is caused by some kind of mutation in the DNA sequence. Now, whether this DNA mutation is something that we can explain or something that we can, for example, if you've been out tanning for 20 years of your life and you get burnt every year, you might have mutated your DNA enough to cause skin cancer. So a lot of the tissues that are commonly dividing, like the skin that divides, you get a whole new set of skin every 10 days, 10 to 14 days. So that's an actively dividing area. Those are most common to have the problems and to have the disruption in the cell cycle. Cancerous cells are ones that are abnormally dividing. And they compete with the normal cells for resources. Usually cancerous cells are dividing more aggressively than normal cells, and so they will beat out. When you have a tumor formed, that's a whole bunch of cancer cells that have abnormally divided. A tumor, so there's types of tumors here. This is the, and I just kind of introduced this is the massive cell, so that's the rapidly dividing cells, and it's forming this abnormal cell growth. If it's benign, if it stays in place, it's benign, and you just remove, a lot of times can remove it, and it's not really a threat. Malignant, the definition is of a malignant tumor is if it has left its original spot. So if you have skin cancer and it becomes malignant, it means it may be in your lymph nodes, it may be in other areas of your body. Okay, so if here's the primary tumor cells, if they are invading and getting into other areas of your body, like your blood vessels here, and here's a secondary tumor, that is when it becomes malignant. In the term angiogenesis, that means the blood vessel growth, literally means like the generation of your blood vessel, so when that grows. Metastasize, that is the name given to the process of when cells are actually migrating to another area. And it's scary. A lot of malignant tumors are ones that have hit, your stage 3, stage 4 cancers. Malignant cells disrupt the function of the normal cells. So functions are abnormal. So for example, if you have a tumor on your thyroid gland that produces abnormal amounts of thyroid, and then you've got other hormonal problems and developmental problems because of that. And that's it, guys. Nice, sweet, short. Hopefully this was a nice review of mitosis and cancer. And I will see you for chapter 4.